Okay, so we're continuing with our journey. We just talked about non-coding DNA and coding DNA. So we have nucleic acids that are made of basic building blocks that are named nucleotides. Okay, the nucleotide includes a nitrogenous base and a sugar and a phosphate. So we're not going to go into the chemistry. You don't have to know the differences between them. Just remember that there's three components and what they are. We're not going to break it down to actual chemistry. Not in our scope. We don't need that. So when we look at the nitrogenous base, and this is important to remember, we have two kinds of bases. We have a family of bases called purines, and we have pyrimidines. Right? We have two of each, so we're going to have two purines and two pyrimidines that are shaped as a ring. So by structure, that ring is connected or linked to a pentose sugar at a different location according to the structure of the base. Okay, such a location on the ring is what we call the prime, and we symbol it as just, that short line. So from now on, that is the prime for you guys. So if you see it connected to the number 5, we will be reading it as 5 prime. So primes are specific locations on those rings. They usually go clockwise, right? So it's easy to remember. The sugars are linked to the phosphate group, usually either at the 5 prime or at the 3 prime location. The nucleic acid is also called sometimes a polynucleotide chain that consists of many nucleotides. Both the sugars and the phosphate groups may differ between each other. They're actually the components that are making the major difference between different chains, okay, and their and the backbone. So the phosphate group connects between the 1 pentose at 5 prime to the next 3 prime. And what you see sticking out of the molecule are the nitrogenous bases. So when we're talking about a nucleic acid, it may have 4 types of bases. The first is represented by the letter A, short for adenine, which is a purine. Then we have guanine, which is a purine as well. We have cytosine, which is a pyrimidine, and thymine, or thymine, which is pyrimidine as well. So we have 2 and 2. From now on, we're more or less going to use the letters. So A, G, C, and T are the 4 types of bases that make up a nucleic acid. Okay? And they're all present in DNA and RNA. In RNA, we find that the T is replaced with uracil, that is represented by the letter U. So the terminal left end of a nucleotide has a 5 prime free group, and the right end has a 3 prime free group. So when we read the upper strand, for instance, of a DNA molecule, it is going from the 5 prime to the 3 prime, and the complementary strand is going from the 3' to the 5 if you look from left to right. Okay? So they're always opposite to each other in their direction. So you have the table, and you can see what bases we're using in DNA, and that in RNA the T is replaced by a U. Okay? And now you're advised to see a movie, so enjoy. So the sequence of the bases is different between species, but also different in their amounts, meaning that it's not as if 25% of a sequence has to be A, and 25% has to be B, and 25% has to be C, 25% has to be T or U. So the order of the bases is different, and also the amount of them is different. Okay? And with time, it has been understood that DNA is a double helix, like we spoke about, formed molecule, which is very, very important to understand the implication of that fact, and we will talk about that. So the DNA molecule is not straight. It actually bends and turns. So about every 10 nucleotides, we have a turn. In addition, when you compare purines and pyrimidines, they're not the same size. Okay? So purines are bigger than pyrimidines. That means that A and G are bigger than C, T, or U. Okay? But they're always placed in pairs, and there's not enough emphasis that I can give you about the pairs. The pairs of the bases are the most simple and yet most important idea, perhaps besides codons that we will talk about in the future, in regards to understanding this whole course. So if you don't understand the base pairs, you'll have a problem continuing. So this is where you stop and start communicating your needs to me. Okay? The pairs are always built by one purine and one pyrimidine. You have no connections between two purines, and you have no connections between two pyrimidines. You don't mix from the same kind. Okay? The proportions between G and C are the same. The proportions between A and T are the same. Why? Because they connect to each other. So if we have a strand, a molecule that has, for instance, 10 A's in it, how many T's are you going to have? The same. If we have a molecule that has 20 C's in it, how many G's are you going to have? The same. Right? When we look at the proportions of G plus C, we actually know the composition of a certain DNA molecule. And G and C can be between 26 to 74%, more or less, give or take a bit, of the whole sequence. Okay? When we want to pair two bases, the way to connect them, this connection in the middle, is done by a hydrogen bond. We're not going to analyze that bond. If you want, you can read about it somewhere. But that's basically the bond that is connecting them. And we'll mention it in the future, but we're not going to analyze it. So We know that G pairs only with C, A pairs only with T, or U if it's an RNA molecule, right? So the two strands of DNA are what we call anti-parallel. One strand, as I mentioned, is going from the 5' to the 3'. Okay, for instance, this strand. And one strand, the anti-parallel one, is going the other way from the 3' to the 5'. Okay? The strands are made out of the bases that are paired between them with the hydrogen bonds. So the phosphate groups on the outside of the chain have a negative electrical charge. That fact itself influences the structure of the double helix, okay, in live organisms, just so you know. When we look at the nucleotides, they are flat and they are perpendicular to the axis of the helix. So if you have some kind of curve in the helix, the bases are going to be structured perpendicular to the helix itself, always. Together, both helixes form what we call a spiral staircase-like shape, okay, molecule, and you'll see that in the movies that you are referred to in the presentation and on YouTube. Okay, so every turn in the helix does not have to have the same amount of base pairs, meaning that we have turns that are longer and turns that are shorter. You don't have to know how many you have in every turn. That's not important for our purposes. Sometimes, when there's too many base pairs per turn, we call it an over-wanded turn. Okay? If it has less base pairs than usual, it's under-wanded. Okay? Okay, so you're referred to a movie, enjoy the movie. Any mistakes in the reproduction of DNA will lead to a different protein produced. Making you make a mistake in transcription or translation, or you have a mistake in the instructions to start with, you're not going to get the product that you want. So imagine you bought something from a store, it comes with instructions, and the instructions are wrong. If you follow the instructions one-to-one, you're going to have the wrong product, right? So that's what we're talking about. Those hydrogen bonds that we talked that are connecting the bases are relatively easy to separate. So that might happen, and it serves us in the needs of translation and transcription. Transcription first, translation second. Let's see. So, since we were talking about the base pairing, and once again, this is extremely important, for every A, I'm going to have what connected to it? A suitable T or U, right? So when I have a strand that has an A, it means automatically that in DNA, it will connect only to a T, and in RNA, it will connect to either a T or a U, right? So one strand is automatically, the instructions determines the other strand. So that's what we call template strands. So one of the strands of the DNA is what we call the template strand, from which we can complement it with its anti-parallel. But once you have the sequence of the template strand, you can immediately know the sequence of the other strand, right? Because where there is an A T or a U is going to come. Where there is a G, a C is going to come to complete it in the other strand. All right, we'll stop here and I'll see you in the next video.